Well, welcome back, everybody. It's nice to be back, isn't it? There's only 300 people here. I think the last time we did this, there was maybe 650 people. But I'm told that we have maybe over 1,000 people online watching this, so we haven't really shrunk at all. We've got bigger. Now, last time I did this, whenever it was, 2019? I can't remember. We changed the format, and the reason for that was because there's just too much stuff now for my tiny brain to hold and then regurgitate live. So what we do is I get the department heads to do most of the heavy lifting now, and I just do a little bit of introduction. And you know the names. Again, if you watch the online talks, you'll know the people. And at the end, I am going to do a little bit at the end, and I've got the luxury now of focusing on things that I think are interesting, which is great, isn't it? I've earned that. 14 years it's taken me to get to that point. So the guys here are going to do the updates that are probably the most pertinent to you in terms of the latest software releases, the latest data, the latest accuracy changes, the latest throughput changes. And there is actually in their slides a lot of juicy detail that we have not yet seen before. I gave a very brief sort of mezzanine update about a month ago, if you remember, online. And there's been substantial progress on what I showed in the interim period. So you need to focus on what they're saying, because those key numbers are embedded in their talks. And then there's a summary slide at the end that tells you where we're at the moment. Again, online, as you know, we have a long library now of previous talks. The most recent one I gave was the Nights Who Say Me. And part of the point of that was to talk about our new ability to measure modified bases. Not just methylated C, but a bunch of others, and there's going to be news on that. And also, I was talking about the relationship between speed of sequencing and throughput. And it's a complicated relationship, and thankfully, Lakmal is going to explain it to you. Hopefully in a very lucid way, and there has been some very significant progress on that front. But I just want to say, Lakmal, you're halfway there. You're just halfway there to the target, right? So whatever Lakmal tells you, it's half as good as it needs to be, all right? Okay, great. So the first talk is James Clarke, and James is responsible for what we call platform, and that means chips, chemistry on chips, and the production of chips and new designs of consumables and various other factors. So with that, I'm going to hand over to James Clarke. Oh, just one more thing. So they've only given themselves 15 minutes, and I've looked at the slide deck, and they've each got about 35 slides. So there's probably zero chance they're going to stick to 15 minutes, which means they're probably going to eat into my time. So when it comes to my time, I might skip past stuff very quickly and just focus on the thing at the end. The thing at the end. All right. Thank you very much. James.
Thank you, Clive. Really nice to be here. As Clive said, I'm going to give you an introduction and an update on the platform. So perhaps best to start with what we mean by the platform. And for me, the platform is all the things that you require to do nanopore sensing. And those are things like the instrument, as Clive said, the ASIC, the electrochemistry, the chemistry that you need to support a nanopore, and in a broad sense, also the nanopore itself, and also the data, the tools that are required to do whatever you want with it. But those areas are better covered by Lakmal and Stu, and they'll be doing that next. I think of the platform as really the base. So we have nanopore sensing. I use that word very deliberately. On top of that, we have nanopore sequencing. On top of that, we have the applications. You heard Dan Turner talk earlier. And on top of that, we have your research. And whenever you are layering something up like that, each layer is adding complexity. And it's really, really important in that setup to have a firm foundation. You do not want to be building on sand. And hopefully, I can reassure you that we have that firm foundation and we take that very seriously. Very, very quickly, because you all know this, we have 3 different types of Flowcell, the MinION, larger PromethION, and our cheapest version, that's the Flongle. And we have a range of instruments to run these, and those include things like the original MinION, very, very accessible, standalone devices such as the GridION, the Mk1D, which is really the go-to for portable sequencing. Those all support the MinION and Flongle flow cells. And then for the larger flow cells, we have the PromethION line, as you know, the P24 and P48. The PromethION chip has really come on leaps and bounds, so we want to make that more accessible, and we have the P2. I'll talk a little bit more about that later. An important point is that we got here by doing continuous improvements and a lot of agile development in the field, and some people really like the updates. There was a number in Gordon's opening about just how many updates we've done in such a short amount of time, but there are people that want us to slow down a little bit, that want that firmer foundation, and for that, we've listened to them. That's the Q line. That is manufactured to very high standards, but we also have a clearly defined and more visible product upgrade pathway. We work on core things, and that's driving towards greater accuracy, more output, more consistency, things like that. Very much kind of core properties which you can build upon. So what have we been doing recently? Clive mentioned his talk in March. That was very, very important because it introduced a change to the Nanopore, the R10 series, which are, as you've heard, the more accurate, latest generation. It's a small change to the pore. Lakmal will tell you more about that, but it's a very big change in terms of what it enables you to do, because it enables you to go as fast, if not a little bit faster, than the R9 series. And for me, particularly, that was the thing that was kind of holding R10 back a little bit, and now we're past that last hurdle. So over time, we will phase out R9, and that makes things simpler for you guys, just having one product offering, and it makes it simpler for us because we can focus on making R10 as good as it can be. So we talked about PromethION output, R10 outputs being very good. Let's put some numbers to that. We have different ways to run R10 kit 14. You heard that from Mitten. The other guys will go into more detail on that. I've chosen the default. That is 400 bases a second, and very similar to the kind of speeds you're used to running on R9. So a good comparison. And we know that Nanopore sequencing is very broad church. That's a great thing. We embrace that. And so I've got 4 different versions here of output. The first we have is really what we use to benchmark. So that's PCR E. coli. I'm sure nobody really cares too much about it, but it's probably a reasonable use case for something like a cDNA. There we got 236 gigabases from that recent Flowcell run. And then we have 3 native conditions. And native, obviously, people prefer because you can get all the methylation, and that's incredibly valuable. So the first is a native human run that you don't have to interact with. You can just load on your PromethION and press go. Very much kind of production mode sequencing. That's 10 kilobases, and we achieved 166 gig from that run. Very, very similar. If you want to go to 30 kilobases, we achieved 155 gig from there. Did come with a few interactions with the wash kit. And for people that really want to have much, much longer reads, we know they are incredibly valuable. Here we had a prep where we had an N50 of 80 kilobases and achieved over 100, in fact 120 gigabases from the output, again using the wash kit. And we really like to see, particularly around this time of year, people tweeting their experience with the latest chemistry. And here you have Matt Luce showing that he got 170 gigabases from his run. So we haven't had R10.4.1 for that long. There's quite a bit we can still do in terms of optimizing the output. So how are we getting these results? One thing that's really helping here is the deeper wells on PromethION, which are in the marathon flow cells. And I've spoken about this a little bit in the past, and now we're making a lot more of these. Very, very simply, the mediator in the well is kind of part of an electrochemical system, and you can think of it like a battery. And the more of it you have, the bigger your battery. And then when you run the Nanopore device, you are consuming some of that battery by passing current. And here we've added more. We've got more volume. That gives you more runtime. And it also, importantly, it gives you more consistency during your run because MinION doesn't have to compensate for all these changes that are going on. So it's only my personal opinion, but I believe that PromethION is the best flow cell we have at the moment. Two reasons for that. It uses the latest generation of ASIC, and that means it is the most accurate. It also has a lot more channels than MinION, and only a little bit more expensive comparatively, so it gives us the best cost per gig. So lots of reasons to be using PromethION flow cells. Perhaps the reason we don't have as many people using it as we'd like is accessibility, and that's what we want to change in the P2. So the P2 can run 2 PromethION flow cells, as you might expect, and we have 2 versions of that. One version is very much like the MinION Mk1D. You plug it into existing compute to run it. You can even plug it into a gridiron. And the other is a fully integrated unit, very much like a gridiron with very powerful compute to run all the base cooling you need. This has been seen in the world. John Tyson has sent a few tweets showing that he has that device. We look forward to following him on Twitter to see how he gets on with the sequencing. If you're lucky enough to be here, it is also out in the live lounge, so please do take a look at it if you haven't already. Lots of unfinished business in R&D, particularly on the platform side. There are always things we want to go and revisit. One of the things that I would like to see change is the flow cell designs, particularly around MinION. MinION has remained relatively unchanged since early access days in 2014. Very versatile, but there's probably more we could do on ease of use, sample loading, etc. On the larger flow cells, you could go to 4-chamber loading. That would work really well on the P2 because you could mate up an 8-channel pipette to that P2. Reducing sample volume. This is something that bugs me a little bit. So nanopore sequencing, you're only detecting what's in or around the pore. You know, it's not a bulk measurement. It's not like the more sample you throw in, the better your signal to noise. It just doesn't work like that. And yet we have 600 microns of fluid in the MinION flow cell above the nanopore, which is a bit of a waste with the amount of material, precious sample you have to put in. We know why that is. It is a bit of a hangover, if you like, from early access days when we design these things, and also how we make the membranes, how we do that wet assembly. So I've got a really good team who are going to completely refactor that, and that will enable new things like flow cell redesigns, but it should also have benefits in and of itself, such as more pores, consistency, robustness, etc. Yeah, we also want to be able to integrate the library preparation. Clive's very keen on that, so I'll let him cover that side of things. Something else that I'm very keen on is changing the electrochemical system. You can see how we have a few kind of constraints there. Silver chloride, another good alternative. It should, I think I've shown before, it lasts longer. It is more stable over time. Here we've shown, the team have worked really hard on this, particularly in the last few weeks. We have shown we can get accuracies over Q20 with that silver chemistry. So they actually managed to get it on a PromethION flow cell, but Flongle is probably where we'd deploy it first, because just that ease of use and that kind of just adds sample. We're really working on the fabrication side. That's where we need the R&D. So yeah, a little bit more about fabrication, robustness, manufacturing. And I put this slide up because we spent a lot of time on this. There are many different groups that all contribute to make sure that flow cell is as good as it can be. And we really get pulled in 3 directions. These are volume, quality, and features. Thankfully, nanopore sequencing is getting more and more popular, and that means we have to make more. That's a very good thing. As we make more, we want the quality to stay the same or go up. We always want to push the quality bar. And that can mean quality to us in terms of our manufacturing yield, which affects how cheaply, ultimately, we can offer these to you. And it also means quality such as how it behaves in your hands, so your outputs. And while we're doing these 2 things, we're also juggling, throwing more features in, and those features can be things like the new pore or the marathon flow cells. And we do this via a method of continuous improvement. When we started doing this in public in 2014, it was a completely different way of doing biosensing. And so we've gone through the cycle, like every new technology will, of monitoring performance, investigating things, characterizing problems, solving those problems immediately, getting everything back on track. And then the most important thing is making sure you capture that problem so that if it occurs in the future, you are robust to it. And that's how we improve things. And why I've put that in there is just the volume of hard work that goes into this behind the scenes. It is that foundations piece, but also, so we've been doing this since 2014. We have a huge 8 years of experience of making nanopore flow cells. So my last section is to talk about kind of where we're going. A lot of people like to hear about new products, and I've said before that a lot of the new products are enabled by the ASIC. That ASIC is like the engine of the Nanopore platform. You can build something around an engine. We have the PromethION and MinION ASICs, but now in R&D, certainly, we have the SmidgION ASIC. And the ASIC does a lot of things, and it's really there to take your signal at your electrode, digitize, amplifies it, pass it through to the base coolers in the end. It also consumes a lot of power, and it is one of the things which, in our current products, can add quite a bit of cost. So we have redesigned an ASIC with really the low-end outputs in mind, but making it as good as it can be kind of from the bottom up. So we've had this in R&D for over 6 months now. I showed this data at NCM back in November, December, and it's the proof of concept data that the ASIC is working properly. This is kind of a shortcut. It's a bit of a hack. We've put the ASIC onto a board and used it in a flongle format. So we don't want this to be in a flongle format in the end. I'll talk about that in a sec. But it allows us to use a flongle flow cell and get some chemistry working on there. We got squiggles, no bells or whistles, so not a lot of data, because it wasn't really running Minnow or anything. But we managed to get enough data out to base cool it. I can't remember which pore this is using, but that data looks pretty nice, certainly good enough for us to feel confident in moving to the next step. So the next step is to build a prototype flow cell using this new ASIC. And there are 3 components to this. We have the ASIC, we have the interface board, and then we have the well array on top, and that's where you have your nanopores. And this is a tried and tested way of building because it's exactly the same format as the MinION. It allows us to access a lot of channels, all the channels on the ASIC as we've designed it this way, and it also keeps the path between the ASIC and the electrode short, and that gives us high accuracy. So really want to do it this way. In the past, the criticism has been that the ASIC is then in your disposable, but this ASIC is absolutely tiny. It's been designed and built for this purpose, and it won't add that much more to the cost. So we're starting to build out a prototype instrument. You can see this instrument breadboard. It is not going to be that big at all. But I do also have a PCB of what is likely to be the form factor of the chip. So it's very, very small. There is an ASIC on the back. This one actually has a flongle adapter on the front, but that will be replaced by the well array in the next iteration. So yeah, this project's coming along really nicely. Like I say, it's the ASIC, very tiny, 5 millimeters by 6 millimeters. A few more details on it. It will be 400 channels. I don't know if I've said that before, but 1 mux, so you get that data very, very quickly. It's incredibly low power. And if you take our best in-field flongle number and you scale it to 400 channels, you get around 9 gigabases, and with those deep wells, I think you'll be able to get 10 gigabases of output from this tiny device. So that's what we're working on, and yeah, I'm going to leave it there. So thank you very much, and hand over to Lakmal.
Thank you, Jesse. So in this section, I'm going to talk about what we did over the last year or so to improve accuracy and output of our system. So most of you know that our system contains like multiple components. We have biological nanopores, and then we have enzyme motors, which controls the movement of DNA through the pore. We have membranes, run conditions, hardware, and software. So usually to improve accuracy, we have to tackle many of these components together rather than one. For example, we improve nanopores base resolution, and then we modify enzyme motors to improve the movement quality. We change run conditions so that we get better defined levels, and then that will help software to base call better. Similarly, to improve the output, we have to tackle many of these components together. For example, we have to improve the motor protein to increase the speed at which DNA is translocating through the pore. And we have to improve the purification methods of pore samples to ensure that we get maximum number of pores in a particular Flowcell. And then we have to improve software to ensure that it detects or define blocking levels of sequencing experiments and so on. So up until last year, our main chemistry was R9 and Kit10. So R9 Kit10 is a very stable system. We have spent quite a lot of time improving that system. And currently, raw read accuracy of R9 Kit10 is about 98.3. And then towards the end of last year, we have introduced Q20 chemistry for the first time. And it contains a new nanopore called 10.4. And it contains new Kit12 contains new motor called E8.1. And together with this enzyme and the new pore and newly improved base caller, Bonito, pushed accuracies, raw read accuracies, beyond 99% for the first time. However, there was a caveat. When we introduced Kit12 R10.4 or Q20 chemistry, our output dropped down because the speed at which DNA translocates to this new pore is about 250 bases per second compared to 400 bases per second of R9 Kit10 chemistry. As Clive said, the relationship between accuracy and output is very, very complex. And usually when we increase the speed at which DNA translocates through the pore, accuracy drops down and so forth. So to improve the accuracy and output together is actually a really hard job. So our teams, however, were determined to improve the output of new Q20 chemistry, keeping accuracy above 99%. So our R&D teams screened hundreds of different pores and hundreds of different enzyme mutants. So far, we have made more than 8,000 different versions of nanopores, more than 3,000 different versions of motor proteins. So in this particular case, we were screening enzyme mutants, changing the how DNA interacts with the motor. We were changing nanopores, the reader head, as well as how enzyme interacts with the nanopore, which means how enzyme sits on top of the pore as well. At the same time, we were changing run conditions. When I say run conditions, we change temperature, voltage, sample frequency, and buffer conditions like salt, pH, and so forth. So in this graph, each point is a combination of a new nanopore, a new enzyme, at a new set of running conditions, temperature, voltage, and so on. So as you can see, towards the end of the speed spectrum, accuracy is high, we can get 99%, but towards the high end of the speed spectrum, accuracy is low. So while working on to improve accuracy towards the high speed spectrum, we picked particular enzyme and particular nanopore around 400 bases per second, at which we get closer enough to Q20 accuracy. And then we trained models at that particular condition to push accuracy above Q20. So that new pore is 10.4.1. And as you can see, if you compare 10.4.1 to the previous pore, R10.4, the speed is improved. Not only that it increased the speed, but it has a much defined or better speed distribution, which helps to improve accuracy even further. And similarly, if you compare the new enzyme E8.2 to previous enzyme E8.1, you can see the accuracy is improved. And this improvement is coming from better defined levels and fewer missteps. So throughout these presentations, when you see this red mushroom with 10 and Kit 14, we are referring to this new chemistry, and you will see that throughout these presentations. So the other nice thing about this chemistry is that you can run the same flow cell and the same kit, but you can tune the speed at which you run the experiment by changing the temperature just using software, and then you can achieve different accuracies and different outputs depending on what you want. So you can see here that our default accuracy, default speed is 400 bases per second. At that speed, you will get, internally, we have achieved 99.2% accuracy. And if you turn down the temperature a little bit to increase the speed, to decrease the speed to about 260 bases per second, you will get much higher accuracy, and we have achieved 99.6. And you can also turn the temperature up and run it a bit faster at 520 bases per second, and then you can achieve much higher output. So which means using the same flow cell and same kit, you can get different accuracies and different output depending on your experiment. And this is for the first time that we have introduced this. Otherwise, we have to have different chemistries. So if you run this in duplex mode, you will hit Q30, around Q30 accuracies. Clive has talked about duplex mode in the last few talks. I will explain a little bit more in the next few slides. And we have released this chemistry to developers, and most of the developers are actually having a really positive experience. You heard earlier from Mithane that he was getting high accuracies, high output. Matt Luce was getting 170 GBs and so on. So this is one of the nicest and smoothest early access releases we have done according to some of our developers. So we were talking about duplex for some time, and duplex chemistry is something that we are working on very hard to implement. In our normal chemistry, we just use single strand or template strand. We let it pass through the pore and we sequence it, and we let the complement strand just fade away. In the duplex chemistry, we capture this second strand as well. So we have seen like about 5% of when we run normal experiments, in about 5% of the time, we have seen that the complement strand actually follows the template strand. And if we capture both strands together and base call as a pair, then we have seen that accuracy increases significantly. But in order to do that, we have to have a perfect sample prep. You need to have both ends of the DNA loaded with the adapters or loaded with the motor proteins, and you can't have nicks and damage in your DNA. So we've been working very hard over the last 6 months or so to improve the yield at which that we get duplex rates. So as I said, we normally get 5%, but we want to be much higher. So we've been, by June last year, we improved the rate to about 20%. So the light blue is a simplex without any duplex, and dark blue is the duplex template, and the orange is duplex complement. Towards the end of last year, we have achieved about 40% duplex yield, but our target is to get at least 70%. So our team is working very hard to achieve that. And this chemistry, duplex is, you can run it both on R9 pores and R10 pores. And as you can see, you can get about Q30 accuracy. And the other important factor is that the accuracy, duplex accuracy, is read length independent. So we are going to make sure that Kit 14 is built in with duplex. So that is another interesting feature. And our algorithms team are also working very hard to improve the duplex base caller. So as you can see in our simplex experiments, we just take one strand and we get the sequence of that. In duplex, we get the template and the complement to get the accuracy. So you can see that Q30, about Q31 is achieved with our current duplex decoder, and which is available in Guppy already. but we are going to improve it much further, and it will be available very soon. So to improve chemistry even further, we are working with a different set of nanopores. So our R9 series of nanopores has a single reader head. R10 series had a double reader head. But even in 10.4.1, the distance between the two reader heads is about 5 nucleotides. So it really helped to improve homopolymer accuracy. So we thought if we can increase this distance between the two reader heads, we can improve longer homopolymer calling even better. So we are working with this new pore at the moment, which has about 9 nucleotides between the two reader heads. By the time we talked about this pore in NCM last year, we had about 90% raw read accuracy. Over the last 6 months, we have improved this significantly, and at the moment, we are about 98%. Still a little bit more work to do before we implement this pore, but this kind of like new chemistry, new pores with much better homopolymer calling is actually coming. So we are also working on other methods to improve accuracy as well. In this particular example, we have introduced random substitutions in our sample prep using PCR methods. So what it does is break the homogeneity of the homopolymer regions. So in red is poly T section, in green is poly A sections. And as you can see in the panel on the bottom left, depending on the number of substitutions you introduced during the, at this homopolymer section, you can see the accuracy improves significantly. This is a single read substitution rate. If you have no substitution versus if you have 3 substitutions, homopolymer accuracy improves significantly. And if you take the consensus of that, the other example is with 70x Medaka polishing. So you can see consensus accuracy versus control is significantly improved. So these are very early results, and we are working on many different aspects of improving accuracy and output. So I hope I gave a glimpse of what we do in R&D to improve accuracy and output. It's a lot more things to come in the future. As Clive said, like we are, well, if we are halfway through, I don't know where we end up when we are 100%, but it's still a lot to do. With that, I think I'm done. I'm going to hand over to Stu to talk about data and where we are with our current chemistry. Thank you.
OK, thanks, Lakmal. So as Lakmal said, I'm going to talk a bit about data, where we are, where you can expect to be with this new Kit 14 chemistry. But before I do this, Lakmal spoke to quite a lot of this. Of course, the critical components of the system, the enzyme, and the pore, and the run conditions that are around that. And Lakmal says, simple, we just change the temperature a bit and the speed changes. But of course, when you change the temperature, lots of other things change as well. So the currents go up if you increase the temperature because the ions are all running around more quickly. And then you may run out of measurement range on the ASIC. You might take the voltage down, keep the runtime. So all these things are interconnected. So any improvement comes with a huge new space of optimizations. And if there's one thing I can say is I know we're not at the optimum condition. That's the only thing I can tell you. We're probably close. It's quite flat. And on the platform, which you heard about from James, we built a lot of headroom into those ASICs in the early days. So we can change things like the measurement ranges and the sample rates. And then when you take the speed faster, you might want to change that sample rate. And that comes with all sorts of knock-on implications for the rest of the system, like the data and the I/O. And these sorts of things will motivate some of the changes we're making deep in the guts of the software. And all those platform things and chemistry things, on the end of that, then you have your base callers, what the architecture is like, what the training sets are like, what's the composition, how you train. All that is connected to get you just to this sort of first first point of accuracy, which is this raw read accuracy. Now, there are several different sorts of accuracy, and the one you're interested in will depend on your application. So raw read accuracy, that's a single piece of DNA or RNA going through a nanopore. We base call that signal, and that gives you your raw read. And you're interested in that if you're interested in single molecules and perhaps a diversity within a population, and you really want that high raw read accuracy. In between that, again, you heard from Lakmal, the Duplex. That's when you can call two reads, the template and the complement. Any more than two, and we're talking about a consensus. And that's perhaps if you're interested in the average of a population. So these ensemble averages, and the few errors that remain in the raw reads, they're mostly averaged out in consensus. But you can ask different sorts of questions. Sometimes you're asking, what's different about this sample compared to the reference? And then we're talking about variant calling. So you've got the SNPs, the single nucleotide changes, you've got insertions and deletions, you've got these much larger structural variations, and we've heard about all those sorts of variants, particularly in Dan's talk earlier. And we also class base modifications as a type of variant. It varies sample to sample, you're comparing to a reference. And then sort of at the end of this spectrum, you've got this sort of test accuracy. That's where you've got a question you want an answer to, yes or no. Is this thing there? Is it not there? Which one of these things is it? And again, we've heard a few talks today where you can get really high test accuracies based on nanopore data. And we showed that ourselves with our Lampore test over the last few years. OK, so where are we now? This is the important question. So raw read, simplex, raw read accuracy, and you heard from Lakmal. I'm going to focus mostly on this default condition, this default 400 bases per second condition. And there we've demonstrated 230 gigabases from a methylene Flowcell, and we're demonstrating a modal accuracy of 99.2%. That's Q21, so beating that Q20 bar. And of course, we do have this choice of run condition. And I think Mitten spoke earlier. He's been running with this, what we're calling the accuracy condition, at 260 bases per second. But in our pockets, internally at Nanopore, we've got this output condition at 520 bases per second. We're still tweaking that a little bit, as it does require changes in sample rate and things like that to get you back up to these high levels of accuracy. But using that output condition, we've demonstrated 307 gigabases from a methylene Flowcell. And then back at the low speed end, the accuracy condition, we've demonstrated 99.6% accuracy. That's Q24. Raw read, simplex. But on to duplex. And as Lakmal said, this accuracy is not a function of read length. So you can have really ultra-long reads with high duplex accuracies. So we're typically seeing with this, I'll just point out, this isn't the default condition, this is the accuracy condition. At 260 bases per second, we're seeing modal accuracies around Q31 there. Our longest Q30 duplex read is 260 kilobases. Our longest Q40 duplex read is 144 kilobases. And our longest perfect read in this data set, which the apps team generated, is 72 kilobases. Perfect. No errors. And what we also see with duplex is the speed at which you run doesn't seem to matter that much. Q29, Q30, Q31 for those three conditions. That says to me there's probably quite a lot of headroom in the algorithms. There's some sort of artificial cap there, probably. And here's what that data looks like. Here's just a bit of a progression of where we've been and where we are now. We've got the simplex R9 on the bottom there. We've got our kit 14 simplex at 400, next one up. Got the accuracy condition, one up after that, and then we've got the duplex reads on top. Very few errors in duplex reads. So now we're going to talk about consensus, and back to my default 400 base per second condition. You get complete, contiguous, and accurate genomes with nanopore data alone. And when you're talking about an assembly, then read length is, of course, a key parameter. When you're talking about consensus, you might be using an assembly, you might be aligning to a reference, and then your depth coverage is a key parameter there. For any of these sort of assembly or consensus or variant calling tasks, we really do recommend Nanopore-specific tools. That's how you get the best results. The workflow here, we're base calling, we're assembling with Fly, and then optionally, these days, polishing with Medaka. The bacterial genomes I'm showing here are just using the polisher built into Fly, so we're not doing Medaka polishing for those, and we're still seeing Q50 assemblies there with pretty modest 10 to 20x coverage, and humans not far behind. Onto variants. So here, really high performance variant calling with Nanopore data alone. As with consensus, Nanopore-specific tools. So when you're calling your SNPs and your indels, our current favorite is this Claire3. So you're aligning your reads with Minimap and then Claire3 to call those variants. If you're doing SVs, it's a different workflow. LRA, long read aligner, and then QtSV, although we are evaluating an alternative workflow based on Sniffles too. And most of these are community tools. These aren't our tools. These are community-developed tools. Claire3, Minimap, QtSV, LRA, and Fly Assembler I mentioned on the previous slide, all community stuff. So SNPs were at 99.9% accuracy. Indels, not far behind at 90. And SVs, and I think Dan mentioned this earlier, SVs are 96%, and we think there's some issues in there with the ground truth that are keeping those numbers artificially low. Ground truth is pretty hard to come by. I will mention we're into the world of bioinformatics a little bit here, so I'll point you to our Epitome Labs product family, where there are workflows and tutorials and data sets there to try and play, and that can really help you get to grips with this world of bioinformatics. And in terms of base modifications, we're still working on the Kit 14 model, so this is the previous Kit 12 stuff. Base modifications, 5-methyl-C, 5-hydroxymethyl-C, 6-MA, all the base modifications, they are built into the Nanopore signal. You're already sequencing them. And now with Remora, this lightweight tool, that is now built into Minno. So modifications are built into the data, and the calling is built into Minno. That's coming out in the next release. And because it's all built in, these visualizations, they just work. So we've worked with the community on this. You can now just see those base modifications directly in your pileups in the popular genome browsers and in many other downstream tools, too many to mention. And as with SVs, when you're talking about base modifications, ground truth is hard to come by. This bisulfite method is not perfect. So we've addressed this, and I think Clive will, if he has time, talk about this later in a bit more detail on these methods, but we have a synthetic ground truth that we've created. And it's on that basis that I think we can say that Nanopore is the most accurate technology for modified base detection. So accuracy headlines, raw read, 99.6% our best number. Duplex, 99.92, that's Q31. SNPs at 99.9. Highly contiguous, I've not spoken about this today, highly contiguous, highly accurate human assemblies. Bacterial, you get circular assemblies from bacteria, Q50 and beyond. SVs at 96, and methylation at 99.8%, the best game in town. We have a few new records. Our Flowcell record is now at 307 gigabases. That's with the output condition. The read length record still stands, I think, at 4.2 megabases, unless anybody's beaten that recently. Our longest Q40 read, I'll say again, is 143 kilobases, and our longest perfect read is 72 kilobases. Now, I just want to highlight a few software features, and then I've got a couple of community notices. Short fragment mode, not short read mode. That's now built into the software. You've had that in Minno for the last couple of months. Nanopore can and always has read any length of DNA. We just used to filter them out in software. So this read length lower limit is now configurable, and you can set that down to about 20 bases if you want, and you can see the sorts of things you can get from, we've demonstrated this with the DNA ladder on that new config. And I'll say it again, accuracy is not a function of read length. And that applies to short reads too. So these are the same sorts of accuracies from short reads as you get from ultra-long reads and everything in between. Adaptive sampling is a feature that is unique to Oxford Nanopore sequencing. And we've spoken about this before, so if you want more details, I'll refer you back to the previous talks. But this is, in brief, this is a feature where if you don't like what you're sequencing, you can reverse the voltage, kick it out, and grab a new strand. And on GridION, we're seeing 5 to 10-fold improvements, coverage enrichment in the areas that you're interested in. This is a pure software technique. We now have this enabled on PromethION. It's not quite where we want it to be, maybe only 3-fold improvement. We've got some firmware things to do to get that up to the levels of GridION and beyond. And that would be as a beta feature in the next MinION release coming in the next week or so. On to my community notices. The main message here is don't panic. Most of you will not need to do anything here. It should be seamless, changeover, happen in the background. But we have a new file format. We're calling it POD5, Pore Open Data. This is a replacement for the Nanopore Fast5 format. I do want to give a special thanks to the Slow5 team at the Garvan Institute and various other places. They've really been really helpful in highlighting and working around many of the problems for downstream analysis with our Fast5 files, which don't scale well to large data sets in these modern computing environments. But we've had to focus on the right performance, because especially if we start to ramp up the sample rate for these faster sequencing speeds, you're running 48 flow cells on a PromethION, you're beginning to butt up against these limitations. And with our file format now, we're seeing a 4-fold improvement in write performance. That's a conservative estimate. We're seeing at least a 10-fold improvement in read performance. So we think we've probably got most of those downstream issues nailed as well. So that's there now on GitHub. Please do check it out. This is a conversation. We want to hear your feedback. We want to get this one absolutely right. So please do get involved. And second notice, there's a new base caller coming. It's called Dorado. This will replace Guppy at some point this year. We're going to get it up to feature parity, and then we'll do the full MinION integration. As with the file format, the design goals here, it's a ground-up rewrite, but here the design goals are high performance. I mean, what we really want is a rapid route from research features out to production. This is open source. It will be user extensible. It's going to support all the operating systems, all the common ones. Multi-platforms, it's going to support the new Apple stuff as well as the NVIDIA stuff. It's built on this the same backend as PyTorch is built on libtorch. So that's there now. That should be live on GitHub. So again, please check that out. Help us out, contribute, comment, try it out. But don't panic. Should be seamless in the background for most of you. I'd like to refer you to the informatics session tomorrow morning, the data for breakfast, where Mike will be talking about Dorado, George will be talking about the file format, Adrian will be giving you a lot more details on modified bases, and Rich Carter will be talking about how you scale up a nanopore sequencing operation. So please do check that out. With that, I'll hand over to Rosemary, tell you a little bit more about our products.
Thank you. Thanks, Stu. Thanks, Lakmal, James. Thanks for covering all of our latest developments in such great detail. It is amazing to be back and to actually be talking to faces instead of talking to a camera. You get really, really good at that over the last 2 years, so we try and figure out if we haven't lost our skills at actually talking to people. So I'm going to cover how we're going to transform all of these latest developments into releases and how we're going to get all of this into your hands. And just as a quick reminder, what we have been building together as a community with all of the tools, all of the amazing developments that you've been doing, is a platform that can perform the full biology from a single experiment. So we've got a nanopore sequencer that can detect SNPs, methylation, structural variation, can do phasing, can do assembly. And we can now go from any read length, from 20 bases, all the way up to the millions and millions of bases that our ultra-long community are generating. We do this while still retaining really, really key features for us, which are simple workflows, scalability, and delivery of data in real time. Another really key feature for us is the fact that we want to scale our platform to your biology. So we don't want you changing the way that you do your experiments because of the box that you need to fit it on. We want to fit to your biology. So from that, we've got all the Flongle, MinION, all the way up to the P48 that can generate up to 14 terabases per run. And we make all of this accessible through starter packs, CapEx-free, so that users can pick them up and start generating data immediately. We've recently entered into a partnership as well with Avantor, VWR, to make the MinION even more accessible and more widely available throughout the world. And we're really, really excited about the P2, and I can see all of the excited tweets that are going on with all of you in the live lounge. So not only do we scale the platform, but we also keep it open, because we believe that biology is diverse, and I think you've been proving to us that biology is diverse through the last few years. So we leave key features of the platform open for you to tweak. So you can choose the number of nanopores, you can choose the runtime that you want to use. Obviously, we've got chemistries that now run at very different speeds. You can run short reads, standard size reads, ultra-long reads, multiplex. All of that is in your hands. And really, irrespective of how you're running the system, what we're aiming to do with all of the open pricing that we have in shop is make sure that you're generating data at really, really, really competitive prices. So the other part that we also do is try and make sure that our upgrades are fully available to the community, so we provide seamless upgrade paths. Through our devices, as we iterate our devices, we make upgrade groups available to our customers. But more importantly, most of our platform upgrades are really contained in the Nanopore, so in the Flowcell, in the sequencing kits that we're shipping, and in the software that's made available. And we have done 221 of these updates in the last 3 years. So we really actually are very grateful for all of you for keeping up with that. Just thinking about the latest release, Lakmal showed all of his conditions that we've been going through and internally. And so we've gone from those conditions in research into conditions in customer hands within about 3 months. We've been able to do this because we have paired up the scale and ability of our teams internally also with amazing compute by being able to rapidly train all of the different models. We've been using the Cambridge One compute from NVIDIA, and that's really managed to about half the time that it takes to train a model to give the feedback back into our R&D teams on whether the condition is working or not. So we are an agile company. We really, really like to put proof of concepts out there really early. We want to hear your feedback. And in terms of speed, I think we've got to kind of thank Mitten. He's gone from flow cells to talk in 12 days. If that isn't fast, I don't know what is. But as James said at the start, we also recognize that there are users out there who want a little bit more stability from the releases. They want to know what they're using, and for that, we introduce this concept of release phases. So we've got developer releases that go out very, very quickly, which then turn into early access, which make all of those new developments available for everyone. And then as we formalize products, we push them into the release bucket, fully released. And for users who really need a frozen platform, we have Q-Line, and we're now working on CE-IVD grade product with our diagnostics team. So now moving on quickly to the releases that are happening. We'll start with sample prep. So on the extraction side, our team continually add new extraction methods onto our community. We've got over 72 protocols up there now. We also have Extraction Central. So that is a place in the community that you can go ask questions of our teams, get support. They're there to help you get the most out of your sequencing experiments. On to long read sample preparation. We'll soon be launching a short fragment eliminator kit. This takes out or depletes reads under 10 to 15 kb from your sample. And we're also upgrading our ultra-long kit. So we've obviously got to upgrade it to the Q20 chemistry, but but we're also adding a lot of different buffers and a lot of different components to make sure that you can work with as many extraction methods as possible. And this kit is with developers now, and we look forward to seeing more and more results from that in the coming days. cDNA sequencing, Dan Turner talked a lot about cDNA sequencing in his earlier talk. So we released the cDNA kit last year. We finally got its sister product out, which is the barcoding kit, and this is now shipping. So we know we've got a lot of demand for it, so thank you for all the customers who've been so patient with us whilst we've been finalizing this one. The teams are also working on a size selection protocol to really address those of you who were really looking at isoform detection. So these kits are all in shop now. You can order them today. On the single cell protocol, a lot of work happened last year. I think a lot of work led by the community. Lebrigand et al., they presented really, really amazing work. When the cDNA kit came out, we simplified that lab protocol there. And the teams today, over the last couple of days, have released Sockeye into GitHub. So Sockeye is a nanopore-only single-cell analysis pipeline, and we're looking forward to your feedback on that. So do go and talk to the informatics team and get more information about how all of that works. On direct RNA, we get a lot of questions on direct RNA. One thing that I was hoping that most of you have picked up on is most of the feedback actually we get is that you need so much input material on direct RNA. So our teams have been doing quite a lot of optimization back in-house, and we have now reduced the poly-A input requirements from 500 nanograms to 50 nanograms. I did hear someone bought a couple of PromethIONs to run just direct RNA, so we hope this helps your samples go a little bit further. And do keep working with us and keep giving us feedback to make sure that we're getting everything you need on that side. So you can't really talk too much about RNA sequencing without mentioning COVID. Our teams continue to support the community with the Arctic sequencing, and of course with the Midnight kit, which is deployed in over 100 countries. We've sequenced over 1 million genomes together, and it's just been amazing to see some of the faces that we saw all the way back in London Calling 2015, when everyone thought this is really, really good for pathogen surveillance and for outbreak management, and it's just been magical to see some of your faces, because we've been working over Zoom with you guys hectically for the last 2.5 years, and we want to really just thank you for all the contributions that you've done. So as we've rolled out COVID sequencing globally, and as it's in all of the public health labs, there's been a bit of a light bulb moment that's been happening across all of these institutions, which is that nanopore sequencing is really good for pathogen detection because of that real time, because of the ease of use, because of that ability and flexibility to respond to what's happening. So our teams are really actively working now with the community to make sure that we're bringing in all of the applications that you guys are interested in, so that we provide a sample to answer workflow, a little bit like the Arctic and the Midnight solutions. And so we've just been working with Amanda Waugh on the African swine fever. There's a lot of interest in pathogen surveillance, be it sort of 16S-based, metagenomics-based, influenza, HIV. And then, of course, our diagnostics team are working heavily on tuberculosis and respiratory metagenomics. So a lot of work going on here. VolTRAX, that's carried on in the background, and what we have now in early access is our COVID sequencing kit, PCR tiling kit for COVID on VolTRAX. So you can sequence the entire COVID genome, you can put 4 samples on a single flow cell, you spend about half an hour and then you walk away. It's a 5 and a bit hour prep, and we think we can do sample to sequence all the way through for about $100. So this is in early access now. We've got customers trialing it, using it in fields, and now that the teams have gotten over this huge hurdle of getting the PCR working and getting it in field, we're now working on the next, all of the sort of the next product pipelines, which include upgrading our range to Kit 14, but going for whole genome PCR, multiplex sequencing, and of course, ligation sequencing kit. One of the really great things that we've actually been really desperate to do for the last couple of years, but we've finally been able to do it, is to get VolTRAX into the flow cell loading zone. So for all of those who are here today in person, you can go in the Flowcell Learning Zone. If you haven't been yet, do go tomorrow and start learning how to utilize VolTRAX. So back to Kit 14, back to the main topic that the guys have previously been talking about. So we're really excited about Kit 14 because from our perspective, it's the best of all worlds. So you're getting the output that we used to get with the R9 chemistry that's been such a workhorse for so many labs, and you're getting the accuracy that we were getting with Kit 12. And the key thing for us to release a new chemistry, of course, starts always with the ligation kit. That is our base kit, the one that gets most utilized, and that is with the developers today. The minute you put a ligation kit out, everyone starts asking for native barcoding. And so the native barcoding kits are very close in our development teams as well. The 24, the 96 barcoding kits, they're all being worked on. It's important to remember that these new kits, they actually contain all of the sequencing components, so you don't need to go and buy extra kits or expansion packs. You just, if you want to do barcoding, just buy that kit. And our teams are currently in the process of getting through 384 barcodes. That's now in our development team's hands. And we will be giving formal timelines for that shortly. But anyone who's interested, please do come and talk to us at the sample preparation tables and do sort of feedback. If you had 384 nanopore barcodes, what would you do with them? From our perspective, it's great. It allows sort of genome sequencing at about $4 a sample. So based on the native barcoding kit, we are going to make a Cas9 barcoding method. So as soon as those native barcode kits go out, the method will follow very shortly after that. As soon as you're done with all of the ligation-based preps, then the rapid-based preps come. Everyone loves the rapid prep because everyone can do them. They're 10 minutes. They're really, really simple. So our rad chemistry is looking really great. Not only will we be upgrading the chemistry, but we'll also be lowering the input to about 100 nanograms. And of course, the RBK kit is also in development. So I know there's been a lot of questions about when can we get RBK, and the teams are actively working on them. And in terms of the timelines, there is an enormous amount to juggle when you're doing a new platform launch. And to try and sort of explain it in a little bit more detail, we've got this slide here. So we're currently in developer access for Kit 14 and R10.4.1. That means a few labs around the world are getting their hands on it, just so that we can check that everything's working correctly. We're then expecting to open the store up for early access in June. And this will be for MinION and PromethION. It will have the ligation kit in there as well. In July, we'll follow that with Flowcell, and we'll start getting the native barcoding kits and the rapid kits out. And as we do that, we will be taking our 10.4 and Kit 12 off the store. And we will be talking to every single person. We know where you live, everyone with a Kit 12, to make sure that you have all the material you need to finish an experiment, or if you want to be upgrading sooner, that we can support you to upgrade to to the latest chemistry sooner. Our laser focus after that point is getting our 10.4.1 and Kit 14 into a released phase, because it is at that point that we will absolutely confirm what's happening with R9, and we want to get it feature complete to make sure that we have a very, very, very smooth transition. So everyone is laser focused on getting to early access, and then we'll be very, very concentrated on getting it into the release mode. So now then to the sequencing part. I'm going to try and summarize Stu's entire talk in one slide, so wish me luck. But what we're doing on the sequencing side is we are raising the bar on our outputs. So you can see here, if we're running all of our platforms at 520 bases, that means the Flowcell could be doing 3.4 gigs, 62 gigs on MinION, 360 gigs on PromethION. We're at 307 now. And of course, as you heard from Clive, it needs to be double. And we've got the short frag mode out now in Minno. So that means that for Nanopore, from our perspective, there is no short read or long read, they're just reads. So we read everything that the platform gets given. We're integrating the methylation, so that's really simple click. And we're listening to the community on our file formats, making all of those better, and of course, getting the new base caller out before the end of the year so that we can really accelerate on all of these latest developments. Apart from raising the bar on output, we're raising the bar on accuracy. I think Stu went through this in a lot of detail, but we are at 99.6 raw read accuracy, and all of our variant calling is performing beautifully as well. So all of these features make it onto our boxes, be it the small portable, so if you want to start exploring nanopore sequencing with a MinION or implementing it on GridION, scaling it on Flongles, so either running more samples or running lower outputs, all of these features are making it through to our small and portable devices, and they are also making it through to PromethION. And with PromethION, we're really, really excited to be introducing P2. This enables people to start exploring PromethION-type scaled experiments, so be it transcriptomics, be it whole human genome sequencing, which you could do on a P2 for under $1,000. We then have a lot of you implementing these in your labs and scaling them up into P24s, and then the P48 just offers that immense scale where users are generating human genomes for under $700, or we are also working with a lot of collaborators on getting 2 genomes per flow cell at under 350. We've had a few already amazing talks on this stage about human sequencing. What we're doing is we've got 3 different types of high-impact projects happening. We've got humans at scale, so we've got installations out there which are running over 2,000 genomes a week, or can have the capacity to do that. We also have the humans at speed, and we heard from Ewan and the rest of the Stanford team today, which, with all of their work, which just completely blows everyone away, the fact that you can get from sample to answer in under 8 hours, and we'll continue to work with them to make sure that all gets quicker and slicker and easier to do. And then we also have human at depth, and we're working with an enormous number of you across lots of different types of, be it cancer, be it rare disease, and all of you are pushing the boundaries of what's known in the human genome. So it's very exciting. So to finish up, we'll look at the analysis. So we have 4 ways of getting analysis pipelines out into the community. We've got our research releases through GitHub, and we really, really value this release mechanism because it really lets us collaborate with all of the tool developers and with all of you who are looking to implement novel mechanisms and novel pipelines. We then have tutorials. So these are a set of tools that we've packaged up. We have a lot of customers who are new to Nanopore. They really want to learn what's going on inside the box. Once we've done the tutorials, or some people want to skip the tutorials, like me, we go straight into labs workflows. So that is everything prepackaged. And if you really want simple bioinformatics, we've got Epitome, cloud-based, single click, and you get your answer. We've been working really hard on the content. The teams have generated an enormous number of workflows, and we support all of these actively. But we've also implemented now a really simple user interface into the Epitome labs. So no more command line needed. If you want to be running workflows either on your infrastructure, your laptop, or one of our machines like GridION, you can now do that with a simple user interface. All of the data that Stu presented, that all makes its way into our open data sets, which is up on Epitome Labs. So anyone who's interested in trying to replicate anything that they've seen today, please go up there and have a look. And so with that, with all of the new sample prep methods, the ways of running, the new improvements in sequencing, all of the analysis, what we hope we're building together with you is that full picture of biology in color. And with that, I'll hand back over to Clive for him to tell you about that one little thing.
Thank you, Rosemary. Who's tired? You all tired? Fed up? They're good, aren't they? They're really good. It's so easy for me to sit there and let them do all the hard work. It is a complicated story, though, right? It is quite complicated. Now, we are making this thing. It is, basically, it's an iPad case. Why would you do that? Well, the advantage of the latest iPads is the new processor, which is, has a built-in GPU. And our benchmarking of that shows it's actually quite competitive. Just by wrapping up a MinION or a Flowcell in an iPad case, we get 5G connectivity, we get cloud streaming, we get a battery, we get portability, we get all the things I sort of wanted on the original MinION, where you shoved it into a laptop, you just sort of fuse them together in a package that has a really good GPU in it. And it is really good, by the way, and very low power. So your chance of running it for a reasonable time and generating a reasonable amount of data, like 6 hours or 5 hours, is actually quite high. So that is being worked on very actively, and you can register your interest in that device right now. That will replace Mk1D. And we will have a pre-order stage. If you have any problems pre-ordering, drop me a line. I love this device. I can't wait for this to come online. I've reserved a little space here on the left. I can't point to it. I've got this sort of idea of having a module there where we can embed a sort of miniature VolTRAX. Because I'm determined to get rid of the lab, if we can do that, and have field sequencing that isn't lab dependent. So let's see how far we get with that. Integrating the MinION is relatively straightforward. Now, you've heard a lot of talk about accuracy, and I think an interesting feature of Nanopore is that a lot of our base calls, especially on the latest chemistry, is actually very high confidence, which is why the SNP calling is so good. It's why the mutation detection is very good. And our errors tend to be confined, as most of you know, because you're here, to the edges of homopolymers and other things like that, that may or may not matter for most applications. So really, it's important for us to be able to really exploit those calibrated base scores when people are doing things like mutation detection. Another aspect of that is we've recently introduced, as you heard earlier, the ability to call modifications. And in Nanopore, it's impossible not to, because in almost every case, if there's a modified nucleotide, we will see a change in signal. It's almost unavoidable. Even a single atom change can change the signal in a nanopore. And even if nanopore A doesn't see that change, a different nanopore will see that change. At the moment, we sort of train over that. We actually blank over modified bases and force the base caller to call them as a C rather than a modified C. Well, that's all changed now. Huge amount of work's been done on actually calling modified bases as they are. As Stuart said, the difficulty is ground truth. The ground truth is really poor. Right? So Adrian, in particular, there's two people who are key for this. One is Adrian Legere, who some of you may know. He used to work at the EBI, and Marcus Stoiber, who's based in California. We believe that our ability to call modified bases is now state-of-the-art. And I'd encourage all of you to go to Adrian's talk and hear what he's done there, and to empirically test that statement yourselves. And here's an example Adrian shows. I'm not going to go into huge detail, but it is empirically testable. We can synthetically create strands where we know where the modified bases are. We can run them through bisulfite, and we can run them through our base caller, and we can quantitatively look at who gets it right and who gets it wrong. And I would encourage all of you to do that as well if you can. Now, importantly for us, the base caller that we use is, as you know, it's a machine learning base caller. And a lot of work's now been done into constructing synthetic training sets. That means we now synthesize DNA. Actually, Nanopore has a program to make a DNA synthesizer that we haven't spoken about yet publicly. That's one for the future. At the moment, we get things done externally by the other companies, but we make randomers. In 1991, I think I programmed my first DNA synthesizer, sort of NNN, ACGT, and so forth. So now we can make a synthetic cassette of random bases, and we can make the 15th base methyl C. And we can construct huge libraries that way, often with sequences you wouldn't see in nature, right? and then we can train the base callers on those, and off we go. And we do very well. And Adrian will cover this in more detail. It's still early days, but our randomer training sets are now outperforming bisulfite training sets. So we're pretty confident. Obviously, we started with methyl C. We're going to now put in all the different modified and damaged bases into those random cassettes, make libraries, and then train the base caller to call any modification on DNA. That's where we're going with that. It's a big program, but the proof of concept looks pretty good. That we can beat bisulfite is pretty good. Now, I mentioned this back in earlier this year. I think the first data we showed back in 2012 that nobody remembers. Obviously, I'm quite emotionally attached to that talk, but nobody seems to remember it. Some of our data from that talk was based on what we call Outy. And actually, a lot of the academic work in Nanopore is based on this Outy method. So what you run at the moment is you run INI. So the motor's on the front of the molecule, and it rams into the pore, and the motor acts as a brake, and the DNA goes into the pore. But what you can do is you can suck in the DNA and have the motor on the other end, so it jams on the other end, and then the motor drags the DNA out of the pore. Right? A lot of our early data was based on that scheme. But we didn't do that. I thought it was a poor design, and blah, blah, blah. Anyway, we are where we are. So we've come back round to it because it does have some interesting features. And now we're looking at a scheme here where we can reread the same molecule again and again and again. That's the main feature. So we can pull a molecule out, and we can make the motor drop back, and then we can pull it out again and drop back. And that gives us a reread. You know what I'm talking about, right? There's another company does something very similar, but we get more than one scan on a given molecule. And that's moving along very nicely. It's still very early days, but the sort of average single molecule accuracy for that technique is pretty much where we were on INI, that's the chemistry you're running, is pretty much where we were about 18 months ago. So it's moving quite rapidly. And there's no reason why we can't improve it further. And I've told the team, the average accuracy doesn't matter, I said. What matters is the per base confidence scores. Now, the way things work at Nanopore is if I say focus on feature A, everybody focuses on feature B. And then feature B comes really good. So you'd think I'd have learned by now to tell them the opposite of what I want. But that hasn't happened. So anyway, it turns out that the average overall accuracy is moving quite quickly. Why does that matter? Well, like I said, our per base calibrated confidence scores can be very high. And that means that, of course, if you have more than one observation, as with the consensus sequence, you can sum the log probabilities. I think that's okay, because when you sequence with Nanopore, the dwell time distribution of the steps is different every time you do it. So I think they're quasi-independent observations. I think it's fine. And anyway, it's empirically testable. You can look at the scores, you can call a mutation, and you can empirically validate that assertion. And that's what they've been doing. And in fact, the example they've chosen, I said, just take a lambda variant and spike it into a lambda, and then sequence them without amplification. And then if you see the variant, how good is the base score? Right? That's what I said. They haven't done that, of course. What they've done is they've taken actual cell-free DNA, they've PCR'd it, and they've measured an actual pathogenic mutation at high confidence in a background, which is what I want this to move towards. What this is all aimed at is that needle in a haystack application for cell-free DNA, where you're looking for 1 in 100 or 1 in 1,000 fragments that have that pathogenic cancer driver mutation in. This is all about liquid biopsy in the first iteration. And the results are so good that they believe they can see PCR artifacts in the amplicons, as opposed to the actual mutations. So it's looking pretty promising. Well, that shouldn't surprise anybody here, because you've all done mutation calling on Nanopore. Right? It's actually very high confidence. Now, I get bled every so often. What a terrible shirt. I get bled every so often. And I'm the substrate for Nanopore research. And the apps team and the development team have done cell-free DNA sequencing. I'm very interested in this whole area of liquid biopsy. And we've done that using fairly standard QIAGEN-based workflows. They extract the DNA, and you can see there's my DNA. It's been fractionated. At the bottom there, you can see the red wine fraction. And at the top, you can see the white wine fraction. There it is. That was a joke. That's an old hematologist joke, but it doesn't play well here. And then we sequence my DNA. And it turns out that we can also, of course, call the methylation patterns on cell-free DNA. And a very preliminary quick look shows that the methylation patterns can be correlated with tissue of origin, which has been, again, tested using an alternative orthogonal method. So it looks like we can not only sequence cell-free DNA, we can sequence modified cell-free DNA. And we can make reasonable inferences about where that cell-free DNA comes from. Very preliminary. This is proof of concept work. It's not meant to be definitive in any way. And then, as I mentioned earlier in the year, we see patterns of size fractions which map or match what you expect from nucleosome footprinting. And the overall size distribution is pretty good. Interestingly, one thing that's different about Nanopore is that we can see longer cell-free fragments. So our cell-free fragments are unlimited because read length equals fragment length. So most platforms have a cap on the length of the cell-free DNA you can look at. In our case, we can actually see longer cell-free DNA right the way up the distribution. Question there, I don't know the answer. Is the longer stuff newer, for example? Don't know. But anyway, this is all just proof of concept work, because all aimed at liquid biopsy. Now, there are a whole number of applications for short fragments, cfDNA being an obvious one, eDNA being another. And as mentioned earlier, we can now do those. It's the same library prep you use anyway. It looks quantitative. We can call the DNA to the same accuracy as very long DNA. And we can call modifications on short fragments. And that cell-free DNA run they did from my blood is now available on Epitome. It's a public data set now. And also, I think on there, not now, but very shortly now, yes, there should be an ultra-long Kit 14 version of me, which I think might be the first human done ultra-long Kit 14. First individual human. So you can go and play with those if you like. And what I would like to do next is a Duplex ultra-long version of me. That's the next data set we'll put up there. So will short fragment sequencing catch on Nanopore? I don't know. It's there. Don't know. I suspect this audience isn't that interested in it, but I think the cell-free DNA stuff is actually quite fascinating. Now, there's a picture of David Cameron there. You know David Cameron, former Prime Minister of the United Kingdom. He's making a really cack-handed effort at loading a Flowcell. And the fact that we have to run Flowcell loading classes, I find, after 14 years, really frustrating. Everybody sort of jams the pipette tip in the hole. And there's that bubble in the end that goes in. Right, so I'm determined to get rid of the pipette. So one of the teams at Nanopore has come up with this thing, which had a very rude name, which we've changed to start with. And it's a little insert there, that plastic thing. Little insert that just fits into the loading hole. And what it does, as you saw, it won't replay, I don't think, unless I go backwards and forwards. What it does is it means you can just squirt sample into that pan, or you can even tip from an Eppendorf into that pan. There's no need now to jam a pipette tip in the hole and squirt and fragment DNA into that hole and put a bubble in there. You can just put sample directly in. So anybody can do it with any kind of pipetting skill, even worse than David Cameron, which is pretty appalling, right? I think that's important. Just a piece of plastic, basically. Actually, it's quite sophisticated. The material, the surface properties, the surface patterning, it's actually quite sophisticated, but it looks like a piece of plastic. Again, I'm very interested in liquid biopsy. So the same team that came up with that has been working on a way to do sample prep on the flow cell. And that means we can put a swab into the flow cell, or a drop of blood, or so forth, into the flow cell, and then everything occurs on the flow cell. That's been a real area of focus, and they're making really good progress. And it's not shown on here, but the way it works is there's a top chamber where the swab goes in, and there's some buffer in there. There's a barrier, which contains a number of things, including gel. And there are 2 electrodes, one on each side of the barrier. And what we do is we electrophoretically move DNA from the input swab into the sequencing chamber. And the other key innovation here, which is going back in time again to the original Nanopore work in 2012, is that we have motors that will grab the end of a piece of DNA. So they'll hop onto an overhang. And those motors are pre-positioned in the flow cell. So the flow cell comes with the chemistry in it. So you just put a swab in, you electrophorese the tiny amounts of DNA into the reaction chamber, they bind a motor, and then they're captured. And what you're seeing here are the usual Nanopore duty time graphs from that system. And it's an Outy system. It's Outy sequencing, it's not INI, so it's the rereading system, naturally. And you can see it's very early days, but from end to end, they can put in a swab of spit or blood, or whatever. They can move the DNA from A to B, the motor binds, and we can sequence or resequence those fragments. That's pretty impressive. I think that's really important. Again, proof of concept. Another key feature on there, there's a button on the flow cell. Again, as James said, we'll probably re-engineer the flow cell. But there's a button on the flow cell that you can push now that will flush the flow cell. So it will just move buffer from a reservoir through the flow cell when you push the button. So you can just flush, 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 flush without having to prepare anything. And all of this is geared up towards moving the device out of the lab and into environments where people don't have vortexes, centrifuges, pipettes, all that stuff, right? Because my vision of field sequencing is you have a little pack, and there's a MinION, maybe a VolTRAX, maybe not. There's some swabs for the bits and bobs, and that's it. That's your field sequencer or your prototype clinical device. So really good progress. This, as most of you know, this is the 3rd or 4th attempt at this we've made. And actually, I think we've got it. I think we've got the design basically right here. I think what we did was we sat down and we decided to think of the ideal device and then make a chemistry that works on the device, instead of what we did previously, which is take the chemistry from the lab and try to make it work in a new device. So we inverted the design criteria. So I'm very happy about that. And again, if you think about it, cell-free DNA, we can sequence that, short fragments, we can sequence it multiply. We have a device where you can put in a drop of blood or a swab. You sort of bring that together, and you're looking at this kind of decentralized, ubiquitized liquid biopsy. And that liquid can be blood, it could be river water, it could be wound pus, it could be swabbing the supermarket shelf, all in one device. That's what I want to do over the next year or 2. Looks pretty promising. There's one other key point I wanted to make. Oh yes, that's right, the toothbrush. So this is where they think I'm completely bonkers in Nanopore. So what does the ideal device look like? Right? If you wanted to decentralize liquid biopsy, how would you do that? And actually, there's some great examples if you think about blood glucose. So early blood glucose, 50 years ago, you'd trot along to a clinic and you'd give 20 mils of blood, and they'd do a huge extraction and run it through a huge thing, and you'd get your blood glucose measurement. And then actually Gordon worked on this, talked to Gordon about it, and then it moved on to strip tests, these sort of lateral flow type strip tests with a reader. And now you can put a device on your arm with a needle in that continuously measures your glucose using tiny, tiny amounts of blood. Think about the evolution of that, it's quite remarkable. And I don't see why measuring DNA or RNA from blood should be any different. I can't see why. So, you know, an ideal device would be a toothbrush, I think, because when you just in the mouth, you've got 50,000 white blood cells per mil. When you scrape your teeth, you release about 100 mils of serum or blood. And doing that twice a day, if you sum over the amount of stuff you're measuring over 1, 2, 3 months, you're looking at 10, 20 mils of blood. You're just doing it in tiny steps. And of course, we can select molecules, we can filter molecules, and if we see a molecule that looks suspicious, we can sequence it again and again and again and be certain that we're seeing a pathogenic mutation. And that all sounds completely bonkers, but that's where I want to take this technology over the next 2 or 3 years. Now, I've missed a bit here. Let me go back. I've missed some slides. Oh, no, they're forwards. I beg your pardon, because I was short of time. So that's the bonkers bit. That's Clive's sort of fantasy product, which is a toothbrush. You clean your mouth, you put it on there, it Wi-Fi's, it goes to epitome, and you've got a trace over time of what's in your blood. And then if you get an infection or a cold or something even more sinister, you accumulate enough measurements to be able to do something about that. Completely bonkers, never been done. I think it's really cool. Now let's talk about protein sequencing, a bit more grounded. So we have a very active program now working on protein sequencing. And this is not the only chemistry that we're looking at, but this is the one I'm going to talk about today. It's important to realize there are lots of ways to sequence proteins on nanopores, some of which have been published. Now, in this scheme, I mean, as you know, we can put a molecule through a nanopore quite straightforwardly, as long as it's charged. But proteins are difficult. They're folded, they may not be charged. Very often they're heavily modified with sugars, all of which are quite tricky complications. And there's an existing technology based on mass spectrometry. It's been there for 30, 40 years. It's the incumbent. You need millions of pounds or dollars worth of equipment, expertise, and the results are difficult to interpret sometimes, quite posterior loaded in terms of what's going on. So we think that the same platform we use for DNA and RNA can be used for protein sequencing, and we're not the only people who think that. So the scheme we're looking at right now is to capture a construct as shown on the left here. And that is actually a double-stranded DNA peptide conjugate. It sounds complicated, but it isn't. And the advantage of that is that we can use the DNA motor that we use for DNA sequencing. Because moving a protein motors are complicated, right? It's been done. We've got collaborators who use the protein degradation complex. But if we conjugate to DNA, we can actually use a DNA motor. And we understand that motor. And one advantage there is that the number of steps the motor takes on DNA is more than 1 per amino acid. Because if you look at the structure of a peptide, if it's stretched out versus DNA, we can actually take more than 1 step on DNA versus 1 amino acid. It's quite an advantage potentially. And so we put a construct on the pore, and it works very much like DNA sequencing, but we let the peptide go through the constriction, as shown in the diagram. And this sounds really simple. It isn't. And then we can generate a signal, and we see a DNA signal, which is quite handy, and then we see a peptide signal. And actually getting that peptide signal has been quite a struggle, but we've got it. A lot of work's been done now if you look at the classical work on DNA sequencing, you'd make a background molecule, you'd make a vanilla molecule, a flat molecule, a homopolymer, in fact. It's not so straightforward with peptides. And then you'd change a base in the middle of that molecule, and you'd move it through the pore. And you see flat bit, flat bit, flat bit, inflection, inflection, flat bit, flat bit, flat bit. So we did something kind of similar with protein. We've taken a very flat, bland, small peptide backbone, and then we've put 1 or 2 amino acids in that backbone, and then run them through the pore to see if we can generate what we need, because the hard problem is generating a sequence-specific signal for a peptide that is reproducible. That's the core hard problem in protein sequencing. And of course, the combinatorics of proteins are enormous, right? You've got lots of amino acids to deal with, combinatorically enormous. But the good news there is that even a not very good sequence can be very unique to a protein. And this is how really mass spec proteomics works. A lot of the peptide interpretations are poor. There's prior knowledge baked in, but they're still unique to a given protein. So in some ways, the bar is lower than DNA sequencing. In some ways, it's higher. It's a very different problem. So in our constructs, we take these vanilla backbones, we change amino acids, and then we concatenate them together. And the only reason we do that is it's just much more efficient to look at concatenamers. And then as you can see here, we're starting now to generate unique profiles for more than one amino acid in a vanilla background. And in fact, it turns out that these profiles are very, very reproducible. Even though they look kind of similar, and some of them look very different, if you look at the two at the top here, it's still remarkable how tight the amino acid pair profiles are. And the average of those is good. So you can generate pretty good tight profiles for pairs of amino acids in a vanilla background. So that's quite, I mean, that might not look like much to you lot, right? But I can tell you that's quite good compared to the state of the art, at least as we know it from literature. It's pretty good. So we're doing well. So what does it lead to? Well, this is not the only scheme we're looking at. I'll emphasize that. We're looking at some other schemes in the background, but we're learning a lot from it. And what we want to do, and this is extremely difficult, right? This is really hard, but as I often tell the team, that's where the value is. That's where the value is. We want to take a complex mixture of proteins from a biological sample, preferably not have to separate them, which is still a key step in proteomics. We want a complex mixture. We can chew them up with a protease, typically trypsin. We have a conjugation method to make a DNA peptide conjugate, and then we shotgun sequence the peptides. Right, so it's not that different from the classical proteomics workflow, but in many ways it's simpler. In classical proteomics, you often have to separate the proteins out first, then break them up. You often have to completely trypsinize the proteins. Our method can deal with partial digests, which means we can have long peptides and overlapping peptides. So there's some really key features here. But ultimately, this is very difficult, by the way. I don't want to oversell this. This is really hard stuff. But ultimately, what we want is any lab biologist to be able to take a complex sample, whether it's whatever, blood, yeast, bacteria, digest that, process it like you do with DNA, and then run it on the same hardware that we use for DNA, so that anybody can do complex proteomics on a MinION or a Promethean or a Gridiron. That's the target of this. We don't need to make a special box. We don't need to make special accessory equipment. We don't need HPLC, so forth. That's what we're driving towards. Very, very difficult. But we think we've got now significant proof of concept that we can generate unique, reproducible profiles of more than one amino acid in a plain background, which is, I've got to tell you, that's how DNA started. Our first DNA work, it was changing a single base in a homopolymer. And then we looked at Kmers and so forth. So the first rung on the ladder we think we've got for protein sequencing. And another key feature is because this is Outy-based, we're using the Outy chemistry I mentioned earlier. It means that we can reread a peptide. Right, and actually, one of the academics published on this, but we've had something like this working for quite a long time. So we can pull it out of the pore, generate a signal, and then drop it back, and then pull it out again, drop it back, pull it out again. And so have multiple goes at that peptide. And then when we're done with it, we can spit it out. So again, this looks promising, but early days. So anyway, so you can see where it's all going. We're trying to develop methods to look at single molecules from blood, spit, urine, water, food. We're trying to make it lab-free. We're trying to do it on cheap devices that we already make. And then we can democratize liquid biopsy, however you define it. That's the basic goal. So with that, I will stop, I think. Thank you very much. Or in fact